Good morning, everybody. Thank you for bearing with me. Here are my disclosures. So we've just had the framework laid out about precision medicine. And precision medicine, is the right treatment for the right patient at the right time, but importantly for us, the right duration. The decisions that you make regarding patients' treatments and how long patients stay on treatment depends on what are now called next generation diagnostics. And within those next generation diagnostics sits imaging. And you can see here, imaging plays a very important role in setting the framework. Now, these next generation diagnostics determine next generation treatments, which you've just had in front of you. So, for example, you can see various types of inhibitors for various pathways. And imaging allows you to pick those, but imaging is also used as a measure of activity of disease, so tumor size, for example, which you then repeat on a number of occasions, trying to find the spot at which you see disbenefit. In other words, saying this patient is getting better, this patient is stable, this patient's getting worse. And hopefully to detect progression at an earlier time point than you would do using conventional techniques. So you can then re-investigate, re-image, and then adapt your treatment. And do that on a number of occasions as the patient progresses along their journey, so that they stay better for longer. And we were talking about 5-year remissions, for example, for imatinib in GIST tumors, for example. And you saw a number of other examples illustrated to you. And so that's the precision medicine concept from an imaging perspective. So in other words, we need a detailed knowledge of what is happening over time. And liquid biopsies, circulating DNA, and tumor markers are not enough for this enterprise, as we've already heard. And so imaging comes in here because it reflects not only tumor size, but also biological aspects of the biology as it sits in the patient, and allows you to look at it over time and see how the patient evolves over time. In other words, we need to take imaging and develop it as a biomarker, which then enables us to look at changing responses over time. So by the time you see this much cancer, the cancer has acquired what are called hallmarks, and I've just listed them on the right-hand side, these hallmarks of cancer. And you will recognize a lot of this: metastatic disease, angiogenesis, the ability of cells to avoid cell death. These are all cancer hallmarks. What we need to remember is that these hallmarks are also anti-cancer targets, and we've just had that illustrated to us very elegantly. The important thing for you to remember is that not every hallmark has to exist in every cancer cell. So you can have a few hallmarks developing and cancer develops. You can have all the hallmarks developing and you can develop disseminated cancer. And the way that the hallmarks develop temporally also differs between different cancers. So there's a degree of biological complexity here. So what we're saying is that as cancers develop, you get cumulative mutations that develop over time, and heterogeneity increases over time. And I just wanted to illustrate heterogeneity in a very visual sense, because we're very visual people, without too much detail. So if you look over here, this is local regional prostate cancer, this is castrate-naive prostate cancer, this is castrate-resistant prostate cancer, and just look at the number of dots. Each of these dots is a mutation. The mutations are arranged according to cellular pathways, so DNA repair, we didn't hear about that today, but we heard about the PI3 kinase pathway today, we heard about the MAP kinase pathway, And if you look as you go from left to right, you see more mutations. So as patients move along their journeys, so the number of mutations increases over time. This is genomic heterogeneity. And if we look just at the top row, just to magnify it up and then give you some idea, if you look at the androgen receptor, in locoregional cancer, it's 2% mutated. Just 2%. It's prostate cancer. It's not mutated, only 2%. But you come over here in castrate-resistant prostate cancer, it's 52%. So you can see how that mutation develops. So we looked at the P10 cellular break, 12% goes to 29%. So this is accumulating mutations on the patient journey. And one of those is the DNA repair defects. And you can see the DNA repair defects at the start turns out to be about 4 or 5%. But in the castrate-resistant state, it's 20%, right? So you guys are going to say, well, what's this got to do with imaging? Well, it's got everything to do with imaging, because you will be able to say, and I'll show you this in a few minutes, think about a DNA repair defect here, because of some of the features you'll be able to see on imaging, because there are therapeutic options. So, for example, you can use PARP inhibitors, you can use checkpoint immune inhibition, you can use platinum chemotherapy for people who develop DNA repair defects. Okay, so finally we are at this point where we're saying, okay, we now understand there's multiclonal proliferation happening for tumors to grow and spread, and that's what we look at. So can you see clonal evolution happening in practice with imaging? So what I'd like you to do is I'd like you to listen to...
So what we ended up building was basically a petri dish.
Listen to this.
Except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. And at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pours a thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, so this was evolution and mutation, which I said occurring in real time over 3 weeks. The real question is, can we do this and see this in our patients? Imagine the petri dish is the human body. The bacteria are cancer cells. Imagine that the antibiotic that you just saw was anti-cancer therapy. Then the light camera, which was used for this, is your imaging system. You should be able to see clonal evolution occurring in front of your eyes. What technique should you use? You should use a technique that shows you tumor proliferation. If you're going to use tumor proliferation, you could use something that looks at cell density or proliferation, and you can use a number of PET or non-PET techniques to this. The simplest one, in fact, is diffusion imaging. Diffusion imaging doesn't look at proliferation, but it looks at the product of proliferation, which is cell density. We know that there is a negative correlation between the ADC value, the apparent diffusion coefficient value, and the number of cells per high power field. That's well established in our literature. The fantastic thing about diffusion imaging is that you can do whole body MRI scanning. In other words, you can get an idea of the full burden of disease, and because it's quantitative, you can look at response assessments. So it's sensitive to how many cells and how viable these cells are. And I just wanted to show you clonal evolution in one patient over time. In this particular instance, we're doing snapshots every 3 months, and the patient is on hormonal therapy. So this patient has a locally advanced breast cancer, and here it is. Here are 2 bone metastases, and we know these are bone metastases because we can go down and focus in those areas on a whole body MRI experiment and show that those are bone deposits. Patient gets treated with just one drug, letrozole and zoledronic acid. So in other words, it's like an antibiotic. And look what happens. There's a good response to treatment after 4 months of treatment. But look what happens between June 2015 and June 2016. This patient is beginning to progress in the breast. Okay, so something happened here, correct? The patient got better, and now the patient is beginning to accelerate. Pictures are quite small. Let's just make them really obvious. Okay, just let's follow this patient. June 2016, this tumor is beginning to get bigger, but look, axillary lymph nodes are also getting bigger. Then the axillary lymph nodes get even larger, the tumor gets larger, and now we're developing liver metastases, and the bone metastases are re-emerging, but at a different location from the original metastatic disease. And then this is the latest scan, and you can see this is now a disaster. Same treatment, followed over time, and what you are watching in real time in this patient is clonal evolution. Okay, so this is the power of some of what you do. The other interesting thing is, remember that diffusion imaging lets you look at cellular viability. In other words, you can look at cell kill, and by looking at cell kill, you can work out how much is alive and how much is dead. And we discussed that in the earlier session. So this is the same patient where we've color-coded what is alive as red, what is dead as green, and yellow is in between, which is, so this is highly likely to be responding, this is likely to be responding, and red is no treatment effect. And what you can see is after 4 cycles of docetaxel, so simple chemotherapy, we can say that the lumbar spine and the left leg, for example, continues to show evidence of disease. Okay, now, when you kill cells, there are several mechanisms, but from a radiological perspective, all you need to do is remember there are 2 ways to kill cells, okay? You can use a shotgun approach, which is like a homicide. So think of that as having a shotgun and going boom and killing loads of cells, and that's typically chemotherapy. What you have to remember is that that is associated with an inflammatory response. When you get an inflammatory response, you get a very large increase in ADC values. So here's a patient with breast cancer being treated with a chemotherapy. We expect an inflammatory response because this is chemo. If you were to look at morphological images alone, you would call this progression. Because look what happens to the T1 signal. You look at the diffusion signal, and it's exactly the opposite. The patient's actually getting better. So we discussed this morning. This is called T1 pseudo-progression. Now, to try to represent that to a clinician, what you do is give them a color map. You say, initially, it's all red. Now your drug works. Actually, you can see a lot of what was red is now green. So a traffic light system. Very effective at trying to display to the clinician that actually this treatment is working like this, but the mechanism, look at how much the ADC changed. Look how it shifted that histogram. You can say that this is an inflammatory response, which explains that T1 pseudo-progression. Now, this is very different to an apoptotic mechanism of cell kill. In an apoptotic mechanism of cell kill, what you're saying is, I'm killing one cell here, one over there, one over there. There is no inflammatory response, right? And so some of the checkpoint inhibitors that we heard about today, a lot of the pro-apoptotic PARP inhibitors, etc., those are killing cells in a very different way. With this mechanism, you don't get that inflammatory response, so you don't expect to see the ADC change. That you don't see such a strong ADC change. So look at this particular patient. If you looked at this particular patient and you looked at the morphological imaging alone, you would say stable, right? You look at the diffusion imaging on the right, you would still say stable, okay? Which would be correct. But the patient is responding. How do I know? The patient is responding because there are areas in the body that have gone green, right? In other words, there are areas in the body that are responding. So showing areas of yellow and green and areas that are still red. So this is very typical of a targeted therapy. So when you start looking at these sorts of images, you can say, this looks like a chemotherapy effect, this looks like a pro-apoptotic effect. Or this drug seems to be working in this way. And you can see heterogeneity of response. And you expect heterogeneity of response. Say, look at what's happening on the left side of the pelvis. The left side of the pelvis is responding better than the right side of the pelvis. And no, there hasn't been any radiotherapy. So the microenvironments are different. And when we see this heterogeneity of response, you know that there's clonal evolution occurring spatially in different areas. If you really want to see a really good example of this, I've got a patient on my YouTube channel where I have done 21 examinations. And it shows you the changes that are occurring over time and gives you a really good idea of clonal heterogeneity. So if you want to really think about this and look at it in more detail, have a look at the YouTube channel. Now, what you've got to remember is that diffusion imaging can be altered not only by cell kill, but also by the extracellular space. So if you were to do an anti-angiogenic therapy and shrink the extracellular space, the ADC value would go down. So you need to think about what your imaging is showing you in relation to the mechanism of action of the drug. Okay, and let me just show you an example. So here's a patient who is being treated with an anti-angiogenic therapy that's very targeted to one particular tumor. So this is using microspheres, using SERT. This lesion was treated with embolization. So in an anti-angiogenic therapy targeted to that particular lesion. And look what happens to the ADC. Look, the ADC goes down. So if you shrink the extracellular space, the cells become more packed, ADC value goes down. In other words, remember, there's a biological mechanism for your observation. You need to understand why that happens. So we were just learning that anti-angiogenic therapies, you need angiogenesis before proliferation occurs. It's a prerequisite. And we know that anti-angiogenic drugs are on the market. You've all got these sorts of images in your clinic. So this is typical for what you would see with pazopanib. The primary tumor devascularizes and the metastases in the liver also devascularizes. So this reduction in enhancement represents the anti-angiogenic action. Now, we always knew that anti-angiogenic drugs don't necessarily cause tumors to respond from a size perspective. So if you look at the table on the right, this is the phase 3 study resulting in the licensing of sorafenib for hepatocellular carcinoma. If you look at the response rates, they are identical. They are identical, right? So why was this drug ever licensed? It was licensed because patients were living longer, right? Look at the survival. The overall survival increased. That resulted in the licensing of this particular drug. Now, so we always thought that time to progression, the time at which tumors progress, would be the surrogate for survival. And it turns out it's not the case. It's not the case. So, for example, if you look at prostate cancer, you look at brain cancer, and you look at breast cancer, for example, yes, it increases time to progression. No, patients don't live longer. Okay, so the inflection point at which patients get worse doesn't necessarily reflect overall patient survival, which is why the overall survival is still an important endpoint. Which is why I'm quite surprised about palbociclib being licensed on the basis of progression-free survival, but that is a different conversation with an oncologist at a different time. Okay, so in other words, we need surrogates, better surrogates about what patients are going to benefit and what patients are going to get harm, and we have a number of these technologies in our clinical hands. So one of the most common technologies that we have is dynamic contrast enhanced MRI, and you know that depending on how you set up the experiment, you can either get tumors going dark or you can get tumors becoming brighter. And you know that when you inject contrast, you're looking at the contrast medium moving from the vascular space into the extravascular space. You can quantify that, and you can do it in the clinic today. So you can work out something called the transfer constant, which is the rate at which the contrast medium moves from the vascular space into the extravascular space, the rate at which the contrast comes back. You can work out the vascular volume, etc. Okay. You need to understand how anti-angiogenic drugs work, because that determines what you see. Let me just show you an example. So here is anti-angiogenic drugs being given to a rat. So you'll say, what's this got to do with me, right? But just look at the pictures. Look at the number of vessels with an anti-angiogenic drug. You see how the number of vessels in the center disappear, but they survive at the margins of the tumor. Do we see this in humans? The answer is yes, we do. Yes, we do. So here is a multiparametric examination looking at diffusion imaging, proliferation, and in this particular instance, looking at blood volume and the permeability of the vessels with somebody who is receiving Avastin, an anti-angiogenic drug. What I'd like you to look at is here. Look, see this area here? Yeah? Before and after. But here on this side, before and after, that doesn't change. So some vessels are being pruned, other vessels are not being pruned. But what happens to the vascular permeability? Areas of red become areas of blue. So there is an anti-angiogenic effect going on. We can see it with our experiments, but we can also see this differential response. That's what the power of imaging, that's what imaging lets you do. And of course, there's lots of other stuff that you need before you see angiogenesis. And one of the things you can see is metabolic changes. Now, there are a number of metabolic changes that occur in cancer, and the same pathways that cause tumors to proliferate are responsible for the metabolic changes that occur within cancers also. Now, we don't have time to discuss in detail the metabolic pathway, so I'm going to skip this. But one of the most important metabolic changes that you observe is something called the Warburg phenomenon. The Warburg phenomenon is where you get increased glucose uptake even when there's plenty of oxygen. Okay, and that's called the Warburg effect, and that results in increased lactate production and is responsible for the FDG positivity because the GLUT1 receptors also increase. So if you look at this patient with breast cancer, you can see this top patient here has invasive ductal cancer. The PET scan is positive, the MRI scan is positive. Look at this patient with lobular cancer. The PET scan is negative, but the diffusion scan is positive. That's a bit strange. What's lobular cancer eating? It's not eating glucose. It must be eating something. It's going to kill the patient. What is it eating? It's eating glutamate. It's eating glutamate. And you can visualize that using something called fluciclovine. But that's, again, a different conversation. So what I'd like you to do is just look at this last picture, which is where we've done a number of techniques over time, looking at spectroscopy, diffusion, contrast enhancement, to give you some idea of the challenges that we now face. So if you look at this patient, again receiving bevacizumab and a chemotherapy, you'll see that the tumor gets larger over time. In other words, the patient's going to die, drug's not working. But if you look at the leakage space, you'll see the leakage space actually goes down, the vascular permeability goes down. That's strange. Is the drug working? Yes, it is. What is the drug supposed to do? The drug is working. It's supposed to reduce vascular permeability, and we can detect that. From an efficacy perspective, is the drug working? The answer to that is no, because despite shutting down the vasculature, the tumor is very much alive. So there are a number of challenges that we face as a community. How are we going to deal with all this data? Multiple functions, multiple time points, all changing with treatment and different treatments. This is where artificial intelligence will come in. That is why you need to pay very careful attention to the next 10 years, because this is where AI will really help you. We need to develop a better understanding of our images. Why does something look like the way it does? What's the biology behind the imaging observations? That's radiogenomics, and we will talk about that also. And what are the therapeutic implications of that? How are we going to deal with heterogeneity? I have no clue, so I'm looking forward to the next lecture, because I think Mu will tell us how we're going to deal with heterogeneity. Are you going to tell us, Mu? Say yes. He's going like this. Say this. No. And what sort of roadmaps? How do we develop a roadmap that enables us to qualify our imaging to meet this precision medicine agenda? Thank you very much.